Good day, this is Professor Wills, CBMD PhD. And today's Tuesday, January 11th, 2022. It is 3:16 and 7 seconds, p.m. Eastern Standard Time. And today's research, liver cancer. So let's hop to it here. New drug combo identified for liver cancer via CRISPR-Cas9 screen. Date, January 11, 2022. Source, the University of Hong Kong. A research team has successfully repurposed an approved drug, drwillphd, a vasodilator. It is to be used in combination with the FDA-approved first-line drug, sorafenib, for hepatocellular carcinoma, HCC treatment. And this study leveraged on their CombiGem CRISPR version 2.0 screening platform 1 to expedite the search among the many possible drug combinations to inhibit druggable targets in the genome for treating HCC. Liver cancer is the 6th most common cancer and the 3rd leading cause of cancer-related death worldwide. Despite the promising initial response to molecularly targeted therapies, tumors are often susceptible to developing drug resistance. It is no exception for sorafenib, the mainstay of treatment for HCC, and the available treatment options are very limited. Drug combination is a strategy for expanding options for cancer treatment to reduce the risk of drug resistance and tumor relapse that often arises in standalone treatment. The simplicity and precision of CRISPR-Cas9 that uses guide ribonucleic acids, RNAs, to knock out any genes in the genome make it a great tool to identify targets for potential drug discovery and development. Utilizing the screening platform of CombiGem CRISPR version 2.0 to generate multiplexable gene knockouts. And this study rapidly characterized the survival of cancer cells following dual genetic knockouts in a pool of cells linked with DNA barcodes specifying the types of genetic alterations they carry, instead of using conventional drug screening array that requires handling of numerous independent multi-wells. And this platform only requires a simple experimentation setup, as a number of DNA barcodes carried by a large population of cells grown in the same culture dish can be counted in high volume via high throughput sequencing technologies, and through screening genes and their combinations from which hits can be translated directly into drug combinations. Using existing drugs, non-conventional drugs, and drug combinations could be discovered and repurposed for treating cancers. And through a combinatorial CRISPR-Cas9 screen, focusing on a set of druggable targets, of which their expressions are upregulated in HCC, cancer stem cells. The research team identified 2 combinations harboring a common target known as NMDAR1, and its parent targets are 2 kinases, FLT4 and FGFR3, of which both their corresponding drug inhibitor is the first line of sorafenib, and specifically genetically ablation of the identified gene combinations inhibits HCC cells growth and self-renewal ability. Based on the Cancer Genome Atlas database, the research team also unveiled the clinical significance of NMDAR1 and HCC, where HCC patients with low level of NMDAR1 expression show better survival outcomes. The team also revealed the enhanced inhibition effect of the corresponding drug combination and their underlying molecular mechanisms. The co-administration of ifenprodil and sorafenib remarkably reduced the cell growth and stemness in multiple HCC cell lines, patient-derived organoids, and tumor xenograft models. Additionally, the team also showed that the unregulation of unfolded protein response, triggering of cell cycle arrest, and down-regulation of genes associated with NNT signaling and stemness could account for the enhanced effects of the drug combination. Ifenprodil has been used as a vasodilator in countries including Japan and France with known safety history in humans, and combined with the first-line sorafenib, the research team has successfully demonstrated that the 2-drug regimen profoundly suppressed ACC cells, growth and self-renewal ability. The successful drug repurposing saves the cost and time that otherwise would be needed for developing new therapeutic agents with uncertain efficacy and safety profile. It also increases the chance of clinical translation for the findings from bench to bedside, as the identified drug combination could be readily tested in future trials for treating HCC. The work has identified a potentially useful drug combination to be further tested for the treatment of HCC patients from approved drugs, which could possibly save or prolong patients' life. The application of the CombiGem CRISPR version 2.0 platform has broadened the scope in search for effective combinations of actionable targets and approved repurposed drugs for HCC in a rapid and simple manner and could be extended to other cancers and diseases. Good day, this is Professor Wills, MD, PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.